All right, so if everyone will mute themselves unless you are the speaker, we're going to kick this off here in just a moment. We thank you for joining us for today's edition of Lunch a la Carte. And we've got some other folks coming in. We'll give them just a moment. So let me begin by saying thank you for being here and also recognizing that a large portion of our supporters and Rotary clubs and Rotarians are still without power and internet and lots of damage going on in our respective areas. And so we recognize that we may have a little bit smaller attendance today, but we are live streaming on YouTube, and we will definitely share this video after the fact. And we know a lot of folks would much rather be here with us today, but our hearts and thoughts and prayers are with all of them, as even in our district here in Western North Carolina, and also our friends in Florida who are continuing to dig out and to deal with a lot that's going on. Just a couple of quick reminders that we do have regular meetings of this event, the Lunch a la Carte, every month, and a monthly call called CART Conversations that happens on the third Thursday of every month. So that is tomorrow if you're with us live today. And we would love for you to join us for that and learn a little bit more about some of the activities that CART has going on. We have some very special speakers scheduled for tomorrow night. We also have a still relatively new online Rotary Club that meets virtually on the third Tuesday of every month at 6 p.m. And it is a cause-based Rotary Club. Their goal is to work on Alzheimer's research. And so we invite you to join them for a call. That was last night. It was a great meeting, but again, their meetings are also on our calendar as well. You can get all of these activities at cartfund.org. And we want to say just a reminder that you're welcome to join any of those and share those with your friends and fellow Rotarians. So I'm going to turn it over to Bill Parker, who is going to introduce our speaker for today.
Dr. Albers, welcome. We are so glad you're here. We had some doubts as we were without water, without power, without telephone, without internet, without cable television. We were beginning to wonder if we were going to make it, but those of us that now have power and everything, we're back on again. Everyone, I'd like to introduce Dr. Mark Albers. Dr. Albers is a neurologist specializing in memory and olfactory disorders. He earned a PhD in organic chemistry from Harvard University, working in the laboratory of Stuart Schreiber, and an MD degree from the HST program of Harvard Medical School and MIT. He was an internal medicine resident for 2 years at Mass General Hospital, and then trained in neurology at Mass General and Brigham and Women's Hospital, where he was one of the first chief residents for the Partners Neurology Residency Program. He completed a postdoctoral fellowship in the laboratory of Richard Axel, where he practiced neurology at the Neurological Institute in Columbia University. At Mass General, he sees patients in the Memory Disorders Clinic and attends on the inpatient neurological wards. As the director of the Laboratory of Olfactory Neurotranslation, his clinical research is focused on developing sensitive probes of olfactory functions as a biomarker for early neurodegenerative disease, including Alzheimer's and TBL and COVID. As the director of the Laboratory of Neurology, Therapeutic Sciences, laboratory research focuses on elucidating the mechanisms and neurodegeneration, identifying novel drug targets that mediate neurodegeneration, and developing therapeutics to prevent neurodegeneration. He is the overall PI of the NADALS trial, one of the first basket trials in neurodegenerative disease. The drug repurposing clinical trial will test the FDA-approved drug, excuse me. Boy, bear with me. Baricitinib in patient populations with either Alzheimer's disease or ALS. He served as a member of the Translational Neuroscience Committee of the American Academy of Neurology, is a core faculty member of the Harvard Program in Therapeutic Science and the Laboratory of System Pharmacology, the Harvard MIT Center for Regulatory Science, and the McChance Center for Brain Health. He is the co-director for the Massachusetts Center for Alzheimer's Therapeutic Science. Wow. Dr. Albers, welcome and thank you for joining us.
Thank you so much. And I'm sorry for that long bio that I shared with you, but I'm very delighted to be here, and I'm really, really excited to share the update. Let's see. Oops, I think I did something. Let me try again. Share. And now let's see. Can you see that okay? No? Okay, great, great. So I'm really happy to report on our progress from the CART grant that we received back in, I think, 2018, 2019. And I have very fond memories of coming down and presenting at the CART meeting. And it was a terrific celebration and great interactions, and just I'm so grateful for the support from the CART Fund, and so glad to hear that everyone that is continuing so robustly. Again, just to echo, I mean, my heart is with everyone, and all of our hearts up here in Boston with everyone that's dealing with Helene and Merlin and you know, all the devastation from that. But what I want to focus on today is drug repurposing in Alzheimer's disease and the therapies, and tell you how the CART grant helped us get to this clinical trial, and give you an update on the clinical trial and where we're going to go from there after that. So these are my disclosures. What's relevant is that I'm going to be talking about off-label use of FDA-approved drugs for research, which is in Alzheimer's disease. And in the clinical trial for baricitinib, Eli Lilly donated the drug for the trial. So I want to acknowledge their support. But as you'll see, the funding for the trial came from both the Department of Defense and philanthropic donors. And so the Eli Lilly did not influence the design of the trial or how it's entirely our lab that's designing and interpreting the trial itself. So what is therapeutic repurposing? And that is when we take FDA-approved drugs and look for new indications. And why is that important? Well, it can help accelerate the new therapies into new disease classes. And so just to give you an example of one, baricitinib, which is FDA approved for rheumatoid arthritis, was then found to be effective in COVID. And in particular, patients that were admitted to the hospital with pneumonia in COVID, they found that baricitinib would reduce the chances of moving from the hospital floor to the intensive care unit and reduced death. And so that was an example of drug repurposing, where they found this anti-inflammatory drug that had effects in COVID. And that's, of course, the drug that we're focused on as well. And I'm not going to have time to talk about it today, but we're also very interested in the relationship between COVID infections, long COVID, and Alzheimer's disease, and also how baricitinib might be useful in patients with long COVID, particularly ones with persistent smell loss, and how that might help reduce the risk of Alzheimer's. But I won't have time to get into that today. But that's an example where this could come full circle. So the other part that's important is that most drugs have multiple targets. So a drug is a molecule, and we think of a drug target. But actually, they tend to interact with multiple different proteins inside the cell. And because of that, then there's a ripple effect. So it's like if you're throwing one rock into a pond and you get the ripples from one rock, But now the drugs are more like you got a handful of rocks, you're throwing it in there, and then all the different ripples are interacting. And that's what we see with how drugs work inside cells. And that's where I was so lucky to be part of the Laboratory of Systems Pharmacology. And what that means is that we look at pharmacology, drug effects at a systems level, not just simply one drug hitting one molecule, but what's the effect on the entire system. And with the advent of new technology and computational approaches, we can now examine those effects and try to then help us determine therapies. And so that's another part where the drug itself, baricitinib in this particular case, can teach us how, the drug works. How is it working in neurons? Is it working the same way as it was originally designed as an arthritis drug? And so it allows us then to not only look for new therapies, but new therapeutic targets. So because most drugs have more than one target, basically they're playing chords on the piano. They don't play single notes on the piano, they're playing chords. And so now can we find new drug chords for Alzheimer's disease? That's the question. And it's really important here, this is data from the 2024 Alzheimer's facts and figures, but it's taking data up until 2017. What we're looking at here is the rate of death. And you see that we've made progress as a field in cancer, particularly in HIV, but the Alzheimer's still remains an unmet need. And one of the things that I want to share with you is that we're taking the principles of the successes from cancer, in particularly breast cancer, prostate cancer, and HIV, which is combination therapies, which has made the huge difference in HIV, and applying those ideas to Alzheimer's disease. So that we think that from cancer, we're learning that it's, breast cancer, for example, is not one disease. It's multiple diseases. And each subset gets a specific treatment. And then from HIV, we're learning that it's probably not going to be one drug that's sufficient, but a combination of drugs or a combination of drug targets that needs to be hit. And so those principles are what we're applying towards Alzheimer's disease. And so this is what motivated our studies that led to the CART grant, which is the observation from my colleague, Teresa Gomez-Isla, that plaques and tangles are not sufficient for cognitive changes. So here what she did was take these mismatch cases. These are people that were cognitively normal in everyday life, and then they came to autopsy. And let me see if you can see that. Yeah. This is a patient that was cognitively normal at the time that they passed away. And you see that they have amyloid plaques and neurofibrillary tangles, and more or less to the same degree as patients who died with Alzheimer's disease. And so, cognitively normal senior, I know it's lunchtime, I'm sorry for this picture, but the cognitively normal, this is a normal brain, and then this is a patient with Alzheimer's. So plaques and tangles are not sufficient to lead to Alzheimer's disease. And so what else is needed to drive that neuronal death that's so critical for cognitive decline in Alzheimer's disease? And so we've been focused on the idea that it's inflammation. That's what Teresa's lab has identified. And what we've shown is that there's increases in inflammation, and in particular, this antiviral inflammation in autopsy brains. So what you're looking at here is each one of these circles is an interferon stimulated gene, and red is up and blue is down. And we're comparing Alzheimer's disease versus normal controls. And you see, these are different brain regions. So this is one brain region, this is a second brain region, a different brain region, all of them infected in Alzheimer's disease. And we see that these interferon stimulated genes are all up. And this is significant at a genome-wide level. So inflammation is important factor in Alzheimer's disease that is important for the cognitive changes in neuronal death. And so again, borrowing this from we think there's different causes that require different kinds of testing that lead to different kinds of treatments. And that's the idea. So how are we going to get to this? Well, that's when we came to the CART Foundation. And we had developed together with, well, our team here, our collaborators here, our colleagues here have developed this, what's called cyclic immunofluorescence, where we could look at up to 60 markers on one slide. So you take a human brain, instead of looking at one marker, which is what I was showing you before, we can look up to 60 different markers. And so we're able then, through this technique, to look at very complicated and ask these critical questions. Why are these patients different? And so that's what we asked funding for. And we were quite successful in taking the funding from the CART Foundation to identify a root cause of inflammation in Alzheimer's disease. And that root cause is what's called double-stranded RNA. Now, double-stranded RNA is genes get turned on, they make an mRNA. That's a single-strand RNA. And then occasionally there's mistakes and it will make then a double-stranded RNA. So that means that the RNA is made from both sides of the DNA. And what we're finding is that the double-stranded RNA gets out of the nucleus into the cytoplasm. And every cell in our body has what's called the innate immune system. And that is an early detection system for viruses. And so essentially, double-stranded RNA in the cytoplasm is a virus until proven otherwise. And the cell mounts an antiviral response. So what we're finding is the double-stranded RNA is starting to accumulate in patients with Alzheimer's disease, about 50% of those, 50% of patients. So we think this is one of the root causes of inflammation. Not the only story by any means, but we think it might be one of the causes. And what we find is that what's really intriguing is the double-stranded RNA is co-localized with TDP-43 pathology. So it doesn't co-localize with the tau pathology in our observations, but with TDP-43. Now, TDP-43 is present in about 50% of patients with Alzheimer's disease. And the Rush group, David Bennett's group, has shown that patients with TDP-43 pathology progress. Their cognitive decline is faster than patients without TDP-43 pathology. And this is how we got quite interested in this idea that COVID might be a driver, because the type of inflammation that we see in COVID and long COVID is very similar to what we see in Alzheimer's with TDP-43 pathology. And these are brains taken before COVID existed, before 2020. So that's where the concern comes from. So with the CISIF data that was supported by the CART funding, we found that the double-stranded RNA is present there, and it leads to this inflammatory response. Okay. Now, with that, then, we're able then to take that idea and put it into a dish. So now we culture human neural cells in a dish, and those are green here in this movie I'm going to show you. And then we tag the double-stranded RNA with a red dye so we can see it. So that's what the red dots are here. Okay. Now I'm going to turn the movie on. So what this is a dish that's in a microscope, and then every 3 hours we're taking a picture. Okay. So what you're going to see is the double-stranded RNA moves down the cell, surrounds the nucleus, the center of the cell. These are neurons, but it moves down, surrounds it, and then you're going to see cells die. They're going to disappear. So, all right, now I'm going to turn on the movie so you can see it, and it should play over and over again. So, again, every 3 hours for 2 days. It only takes 2 days to kill the neurons. And so this will play again. Let me show you this here. If we look at this cell right here, for example. So there's the double strand all around the nucleus, and now the cell's gone. So we can use this double-stranded RNA. We see that in human brains, and now we're seeing it in the dish. where we get cell killing of neurons. Okay. So the double-stranded RNA is present in 80 brains, and it's sufficient to kill human cultured neurons. So what's happening here? This is a cartoon of what we think is happening. So here's the double-stranded RNA in the cytoplasm. It's recognized by these pattern recognition receptors. These are the detectors for viruses. That's how they evolved. They're viral detectors, and they detect this double-stranded RNA. So that leads to the production of interferon. And interferon gets secreted. That's one inflammatory marker. It then binds to its receptor on the same cell and neighboring cells, and it triggers this cascade. And these interferon-stimulated genes, ISGs, there's 2,000 interferon-stimulated genes. I showed you some of those that were upregulated in Alzheimer's disease. So that's what we think is going on when we have the accumulation of double-stranded RNA. So here's a subset of those 2,000 interferon-stimulated genes, and in these different regions. And it's just showing you when it's red, it's up, okay? So it's upregulated in multiple different brain regions, these interferon-stimulated genes. So here, I guess it's 354 different genes that are upregulated. And so that's a big response. So here's what we think is happening. As we age, we start to accumulate cytoplasmic double-stranded RNA. And somehow that's related to the TDP43 inclusions in about 50% of patients with Alzheimer's. We're actively investigating what is the source of the double-stranded RNA. It's largely coming from the genome. It's largely coming from our DNA, not from viruses, is what we're finding. And there's several elements. So there's genomic instability as we age. There's every day in our cells, each one of our cells in our body, there's 1,500 double-stranded DNA cuts every day, and those get repaired. Well, in neurons, because the neurons in our brains are just as old as we are, though they accumulate that kind of damage. And occasionally, what happens if the two cuts are near each other, when it gets stitched back together, it gets inverted. And now all of a sudden, one chromosome makes one strand, the other chromosome makes the inverted strand, and those two find each other and can make double-stranded RNA. That's one idea. Within our genome, half of our genome is comprised of what's called transposable elements. These are, through evolution, we accumulated these, and many of them have inverted sequences within them. And one of TDP43's jobs in the nucleus is to suppress those so that they don't get expressed. If TDP43 comes out of the nucleus, then we see de-repression of these transposable elements. And so they start getting expressed, they make the double-stranded RNA, they bind to each other, and then they can get in and cause the inflammation. And the third is viruses. There are viruses that can get reactivated, and that could lead to double-stranded RNA. The innate immune system is agnostic. It doesn't recognize the specific sequence. It just recognizes the structure of double-stranded RNA. So it doesn't know what the source is. So in any event, with that, you get this interferon signaling that's quite robust, and that leads to neurodegeneration. So at the present time, we thought this seems very complicated, but there's a funnel through type 1 interferon signaling and then to neurodegeneration. And so could we then use that to block this activation of microglia, which you've probably heard from other speakers, these reactive astrocytes, which you've probably heard from other speakers. And so we want to target that type 1 interferon and see if that can block the neuronal death. Okay, so I showed you in the movie before that double-stranded RNA can lead to the type 1 interferon and neurodegeneration. So now the question is, will this JAK inhibitor, will this particular drug that's called a JAK inhibitor, but that blocks this interferon doesn't rescue neuronal death. And so the short answer is yes. So here is baricitinib. And what we're looking at here is cell viability. So with no baricitinib, we get killing of the neurons, and the same neurons that we saw in the movie. And as we increase the dose of baricitinib, we get rescue of the neurons. This is ruxolitinib, also FDA-approved drug. This is approved for cancer. Again, we see dose-dependent rescue. This is tofacitinib. This is also FDA-approved JAK inhibitor from Pfizer, approved for rheumatoid arthritis. It also leads to a dose-dependent rescue. And so by blocking the interferon signaling with these drugs, we were able to rescue neuronal death. That was in one kind of neuron in the dish, so we checked another type of neuron. We actually checked now 4 different types of neurons in dishes because we want to be careful. But here's a second one, just to give you a sense of it. Again, here's baricitinib in a dose-dependent manner. Here's the killing that we see, and then we can rescue that. In this particular cells, these are cortical-like neurons. So this is the most relevant neurons for Alzheimer's disease. These are like in the cerebral cortex. And we get almost, basically full rescue at a higher dose of the drug. And so we were excited about this. If we can find the right patients that have this kind of inflammation, then maybe that can help rescue their neuronal death and see if that can slow down the disease. So how is the drug working? If we take cells, these are neurons again. If we add the double-stranded RNA, then we get an upregulation. You see the activation of this particular JAK protein that was on that pathway. And then if we add either, this is without the double-stranded RNA with baricitinib alone, and then this is the real experiment here. Baricitinib blocks that phosphorylation, so it blocks the inflammation. Ruxolitinib does the same thing here. So we can block the inflammation inside neurons with these cells. So we rescue the inflammation and we rescue the cell death. So baricitinib is an FDA-approved drug. It's FDA-approved for rheumatoid arthritis, and now it's also FDA-approved for COVID. And recently, it got FDA-approved for alopecia, so hair loss, for inflammation for hair loss. So this drug seems to be doing a lot of good things. It also has some, because it's so potent, it also has some side effects. In particular, it can lead to increased risk of pneumonia. Even though it's approved for COVID, it can block the ability or offend, like when you're trying to fight off a pneumonia, it can make it more difficult. All of our patients in the clinical trial have to have this shingles vaccine because it can reduce the reactivation of shingles, and it can lead to difficulty with blood clots and malignancy. So our immune systems are fighting cancer all the time. If we block that immune response against cancer, that can increase the risk. So the FDA made us, in the clinical trial, we could not have anybody with a history of cancer in the clinical trial. And so with that, what I told you, we started with the observation in the brains from the CART funding, the inflammation, finding this drug that blocks it. We were able then to design this clinical trial. And so what we have done, or the way the clinical trial is designed, is everybody gets the drug. There's no placebo. But what happens is you come in, people who are interested come in and they get a spinal tap at the beginning. We then test at that point, do you have this inflammation in your spinal fluid or not? If you don't have it, then we say, this is not a good drug for you, and we don't think you would benefit from this trial. So you're not invited to continue. If instead you do have the inflammation, then you're invited to continue in the trial. And so we had approximately a 50% screen fail rate. So what we kind of predicted from the TDP-43, it's what we found in our Alzheimer's patients, is that some had the inflammation and some did not. And so that fit, kind of our expectation. So we had planned for that. The patients that we let in, or the patients that were eligible for the trial, were subjective cognitive decline, so pre-MCI, MCI patients, Alzheimer's patients, even to moderate stage dementia. So we were fine with that whole range. And so what happens, the people who are invited to continue, they wait 8 weeks and they get a second spinal tap. Then they start the drug at 2 milligrams. They go 8 weeks, they get a third spinal tap, and then they go on 4 milligrams of the drug, and they then get another spinal tap. So what we have at the end is 8 weeks apart, no drug, 8 weeks apart on 2 milligrams of drug, and 8 weeks apart on 4 milligrams of drug. So we can compare in each individual, did the drug help your inflammation, and did it help your neurodegenerative biomarkers? That's our question. And then, so we can look at each person individual, and then we can pool them together. Now, the reason it's a basket trial is that we find the same mechanism in ALS. So ALS is a motor neuron disease that affects predominantly a little bit younger population on average, and it's different from Alzheimer's because males are affected more than females. But we think that at the molecular level, it's the same phenomenon. And if it happens to act up, this unusual event acts up in your neurons in your spinal cord, you get ALS. And if it acts up in the neurons in your brain, you get dementia. But it's kind of the same gene. Again, that's akin to cancer. There are oncogenes that if they act up in one particular tissue, you get, for example, liver cancer. And if they act up in a different tissue, you get lung cancer. But at a molecular level, it's the same mechanism. And so we borrowed this idea from cancer. A basket trial in cancer is the two different diseases, such as lung cancer or liver cancer, but the same oncogene and the same drug. Here, what we said is there's inflammation in the spinal fluid of both types of patients. And so we think the mechanism might be similar. And so we wanted to look at both patients with ALS and patients with Alzheimer's disease. And so that's what we have in this trial. Now, the trial is funded, the ALS side of the trial is funded by the Department of Defense, and the Alzheimer's side of the trial is funded by philanthropic, grateful patients, philanthropic donors to fund this part of the trial. And so the philanthropic work is so important for this whole effort. And then, as I said, Eli Lilly donated baricitinib to the trial. It is an expensive drug, so we're very appreciative to Eli Lilly. It costs about $6,000 a month to take it in the US, and it's a pill, and you take it once a day. But right now, insurance wouldn't cover it for this reason, for looking at neurodegenerative disease. So we are grateful to Eli Lilly for their donation. So what are we trying to do in this trial? 2 questions. One, does baricitinib get into the nervous system? It was designed as an arthritis drug. How well does it get into the brain? So that's one. And then the second is, does it move the inflammatory biomarkers in a significant way in the spinal fluid? And so these are the 2 questions that we want to ask. In addition to that, we have other we have other markers of inflammation that we're looking at in these secondary objectives. So the names and the details are less important, but these are all inflammatory biomarkers. And the second thing we wanted to ask is, does it block neuronal death? Like we're seeing in our cell lines, and I didn't show it, but it also rescues neurodegeneration in our mouse models. So does it rescue neurodegeneration by this NFL, neurofilament light chain, which is a marker of neuronal death, tau, phospho-tau, and does it change TDP-43 levels in the plasma? Then we also want to see, does the concentration of the drug correlate with the CSF changes? So do we think that it's causal? You know, if you have a higher dose, do you have a greater effect? And then finally, we're also trying to, of course, find out if it's safe or tolerable in this patient population, because of course it was in rheumatoid arthritis in the initial clinical trial. Now, what's not on here, and what I'm also extremely excited about, is the exploratory objectives. And we have funding so that when the trial is done on all the patients, for all the samples, we will be able to look at the proteomics, so all the proteins that we can measure in the spinal fluid and the blood. And so we can then look for, these are ideas that we had, but maybe there's others that we're not aware of that could teach us how are we best able to select for patients that might respond to baricitinib. And the second thing we're doing is we have funding to take all of the peripheral white blood cells. So we draw the blood, we save the cells, and now we can do a technique called spectral flow cytometry. What that allows us to do is 42 different markers on the cells, so we can separate them out into different classes and then see what does baricitinib do. So we think that there are signatures for the different types of diseases. And so then with a blood draw, the idea would be we would be able to determine whether a person was a candidate for further treatment with baricitinib or not. That's what we want to learn. So that if this trial is successful, then we could move forward with a larger trial where we design it so that the specific types of patients that have this type of inflammation would be the ones brought into the trial. So those are the goals that we're seeking in this clinical trial. So I'm happy to report that as of September 30th, we've finished our recruitment phase. So we have all the patients in the trial now, and we're expecting the last patient to get the last dose of baricitinib sometime in May 2025. And so therefore, we hope that the results of the trial will be starting to become available in the fall 2025. It'll take us a while to get them all analyzed. But this is the long-range goal of being able to report that out. Now, there's a couple of things we're doing other than that. And so we've also started to learn, how does baricitinib work inside those neural cells that I showed the movie of? And we actually believe that the way it works is hitting two different proteins. Sorry. Sorry, I'll keep my hands down. Two different proteins. Neither one of them are JAK, or neither one were the target that the drug was initially designed to hit. And so we're now very actively exploring those two different molecules, or those two different drug targets, and seeing if we can come up with newer drugs that are more specific. Because then the hope is that we might be able to reduce the side effects with baricitinib that we see with baricitinib, because baricitinib is a very powerful drug, and we might be able to have something that's a little bit more on target. And so baricitinib, I think, could be an early, has potential to be an early, because it's already FDA approved, but also open up a whole new area of drug targets that we could use to come up with more specific therapies in the future. The other thing we're doing that I wanted to talk a little bit about is for the last 8 years, separately, I've been leading an effort to emulate clinical trials within the electronic health records. And actually tomorrow, we're having a meeting here that will involve investigators coming from Israel, coming from the UK, and then several places around the US. So we have our center here in Boston, collaborators from New York, collaborators from California, and we're hoping to recruit the one Florida group. So that would be the electronic health records for many of the patients in Florida. And because baricitinib is an FDA-approved drug, in addition to doing our clinical trial, what we want to ask is if we look at rheumatoid arthritis patients that were treated with baricitinib, what's the rate of getting Alzheimer's disease in those patients compared to patients that were treated with other rheumatoid arthritis drugs? And so that's what we're working on. We call this a target trial, and we're emulating that in the electronic health records. We need because baricitinib is not yet, we don't have enough people just in our Mass General Brigham electronic health records on baricitinib to run the trial effectively. And so the meeting tomorrow what we're trying to do is work together so that the data, the electronic health record data stays at all those different places so that the privacy is concerned. Each site then runs the trial and sends the results to a central place here in Boston. We iterate across all those sites, send back the information, and then they can send it back, run it again. And so we eventually converge as if we had everything together. So we can preserve the privacy, but we can do something where we get enough information to run the trial. So we're very excited about this as an augmentation therapy. And we've been, as Bill mentioned, I'm part of the regulatory sciences program here at Harvard MIT, and I had a fellow who was working on that as well, how to work together so that maybe one clinical trial plus an electronic health record emulated clinical trial could be the data that's needed to get the new indication for a disease such as Alzheimer's disease. So we're exploring this in parallel with the clinical trial. All right, so I have many, many people to thank. This is a team effort, and this is the team that's largely involved in the laboratory research at Mass General and at Harvard. And in addition, these are the people that are doing the electronic health record trial work here. In the same time, and then these are my funding sources. And again, I thank CART for their the grant that got all this started, and or that definitely helped us get this started. And happy to report this progress, and I'm happy to come back in the future after the clinical trial is done and report back to the CART team. So I'll stop here and happy to answer any questions.
Greg, you have a question.
Dr. Albers, thanks so much. I mean, it's a hugely complex topic, but you really helped bring home some of the main points. This is the third one of these lunch and learns that I've been on, and each research project focused on a very different possible cause and possible trial cure of, or at least some sort of impact on Alzheimer's. How much does your work get used by other researchers in saying, okay, well, if he's doing this, maybe I can start taking a little piece of that and going over here and seeing if that's something that's important. Does that make sense?
Absolutely. No, I think that's so that's so important. That spirit is so important in tackling this disease. That we want to use and leverage the expertise from other investigations, because I believe that it's going to be combinations ultimately, that none of us are going to have the magic bullet. I think it's going to be a combination of different therapies and chipping away at the different in each individual patient. I don't think there's going to be one cocktail for every patient in my clinic. I think it's going to be, all right, you have memory symptoms. We need to investigate in you what's happening and then get you on the right therapies for that. And so, absolutely, I think that we need to continue this collaboration. I mean, these communications and collaborations, so that we incorporate all of that. We're just to give you an example. Part of the reason that we're looking at tau, which I'm sure you've heard about from other speakers, is that there's Bess Frost is somebody who's identified double-stranded RNA accumulation with tau pathology. And so that's why we're looking at that. We borrowed, we saw her results, we found it interesting, and then we want to, explore that connection as well. And there's multiple types of things going on all the time with regards to both people within Alzheimer's disease and also outside Alzheimer's disease. And there's connections that we're making in the cancer field in particular. It's quite interesting that way. If you don't mind me taking a minute on this, if you look at the epidemiology, if you look at the disease associations, people with Alzheimer's disease have a decreased risk of cancer. People with cancer have a decreased risk of Alzheimer's disease. And so it's kind of a curious, inverse relationship. If you look at the inflammation and you imagine that, we inherit with our genes that we've inherited, we all have a thermostat on our inflammation. And you can imagine if it's turned up a little bit, then you're very good at killing cancer cells, but maybe you're at a higher risk for neurodegenerative disease. If your thermostat's turned down a little bit, then you're at less risk for the neurodegeneration diseases like Alzheimer's, but maybe you're not as good at killing the cancer cells. So we're very much looking at how do cancer cells suppress the immune system, and can we use that and harness that knowledge, as the cancer investigators understand that process, can we harness that and use that as a therapeutic approach? And so I completely agree with you. I think it's very important that we stay, in contact, not only with the people in our field, but also outside our field.
Well, I certainly appreciate the explanation. A really terrific program. I wish you the best with all the research and the results that you're expecting. I appreciate your working so hard at this disease. Thank you again.
Thank you. Yes, Bill.
Dr. Albers, your comments about ALS really strike to the heart. My wife's sister died at a very young age with ALS, and we have really been attuned to any research involving ALS causes, cures, whatever, and it's all been so negative over the years. This is the first positive stuff that we have seen about ALS, so thank you for that, as well as dealing with Alzheimer's disease. This is terrific. I really appreciate it.
Well, thank you so much, and our fingers are crossed that maybe we can help make a difference a little bit, at least, and move in the right direction.
I may remember this incorrectly, Dr. Albers, because that happens quite often with me, but I believe Drs. Kim and Quinty are also at Mass General with you and received a CART grant a couple of years ago now that are also working on the testing of repurposing drugs. And so it's nice to hear that there are a lot of different options that you guys are looking at right now.
Absolutely. So Dr. Kim's lab is upstairs from mine, and we're in close contact. In fact, I was emailing with him last week on stuff. The work that he's doing, I mean, the model system that he's developed is tremendous. And some of the hits that he's found are very similar to what we've found. So we're working together on those. In addition, he's found different hits that I'm very excited about, and I'm trying, I'll give him my advice to help, in terms of moving forward. But yeah, absolutely. It's a tremendous, I'm very lucky to be here and be around so many great scientists, and thank you for supporting him because he's terrific. So made a good choice. He made a good choice.
Anybody else have questions, comments, thoughts? This has been really fascinating. Thank you so much, Dr. Albers. This has been great.
Well, again, thank you so much for all that you do. And the philanthropic support is so important for us as scientists, because this is how we get to be creative and get the data generated. I mean, you see like that grant and literally millions of dollars that have come from the initial grant that you provided. And that's what's so important about what you guys do. And I really, really appreciate your efforts, and anything I can do to help, please let me know.
Thank you. That's what we like to hear, that these seed grants are effective and are helpful. And we hope that you will encourage others from Mass General and others to apply. This coming year, I don't know if you heard, but in 2024, we did give away a record amount this year of $1.4 million.
Wow.
And we are looking at doing even more next year, hopefully. So stay tuned for that.
Amazing. Thank you. That's wonderful to hear. Much, much appreciated. Yeah. Thank you. Yeah, exactly.
So we thank everybody for being here today. It is always a different date every month, as you know, for Lunch a la Carte. We try to be cognizant of the busy schedules and different time zones sometimes of our researchers. And we definitely appreciate them taking the time to prepare for a very different audience from what they're accustomed to presenting to. We recognize that they are used to being in front of peers and other medical professionals, and we really appreciate you making